we begin implementing an aggressive hygiene routine. We start removing worms. And lo and behold, we get all of these diseases, allergic disease, autoimmune disease. So is that a coincidence? Probably not.
Hey everyone, I'm Karl Schmieder, the co-host of Grow Everything, and I'm here with my host, Erum Azeez Khan. Erum, what is going on?
Hey, Karl. Yeah, it's going on. Things are super, super fun and busy. So you audience, you're going to be listening to this right after the Biofab Fair, which is Biofabricate's new unconventional conference that Suzanne and Amy have worked really hard on to change a conference vibe, make it a little bit more of a call to action versus just people just listening. It's like, okay, no, we need to have a bigger conversation about how we can build the bioeconomy, especially in the lens of fashion, design, personal care and beauty, construction. Very exciting stuff. And at this conference, we recorded our 150th episode in front of a live audience. I wanted to say live studio audience, but it's not really a studio. But hey, whatever. How exciting, huh, Karl?
Super exciting. We are recording this before our travel to London, and you'll listen to this after we get back, and that will be our 150th episode. But it's event season. September is the season where everybody tries to get as much work done before the end of the year holidays. So we have been attending events around New York City. Erum, you were at an event. We actually were both at events in Queens. We don't usually go there for events, but there is a number of new lab spaces there. You were at an event at InnoLabs. Do you want to talk about that for a minute?
Yeah, absolutely. And it's funny you said Queens because I would say all the real estate people and the people that live there will like to call it Long Island City. But it's true. It is Queens. So, hey, I don't know. I was born in Queens. I love it. And it's not as far as people think to get there. It's actually pretty easy. So everyone relax. If you're ever invited to Long Island City, just go. Don't be like, oh, it's so far away. Give it a try. And yes, this event, it's called Made in NYC, Built for the World, Science, Startups, and Opentrons. And so this was a very exciting panel discussion hosted by Nucleate, by Biolabs at NYU Langone. Opentrons was there, and it was sponsored and moderated by Millipore Sigma. So a lot of heavy hitters up and down the value chain. And wow, I'll have to say that this was a great conversation. It was definitely rah, rah, rah NYC, but it is true. Just the amount of information that was shared, like Opentrons starting in GenSpace.
Yeah, they started in GenSpace. Yeah, and we'll talk about that in a minute when I talk about the event I went to.
Yeah, so just people coming together and they were like, hey, look, I can't afford some of this really advanced lab automation equipment that's being sold by these big industry players. How can I just put together some pipettes and the 3D printing tools and figure this out myself? And two people came together and they figured it out. And there was a lot of talk about the challenges they encountered and everything, but they saw a market for a low-cost device. And they thought it was just going to be like biohackers and like people that are like niche, small community people that would want to play around. But then they saw a whole market for hospital systems to buy this lab automation to do different types of tests and experiments, and universities. And it just blew up. And what a great success story coming from brilliant minds, happen to be located in the New York City area. Brooklyn in particular. Hello, shout out to Brooklyn in the house. So that was great. And then Glennis of Biolabs, although she has moved on to another organization, I just love talking to her. She's just tells it like it is. She's very specific in her conversation. She comes with data. She comes with numbers. She comes with real advice and insights that people can take action on. So anytime I hear Glennis talk, and we had done an episode with Glennis Mera of Biolabs, or formerly of Biolabs, so please give that a listen. And it was just great to see all of our friends. InnoLabs is newer space. It's beautiful. They've got a lot of people to move in. Karl, I wanted to share that with you. I don't know if you knew that, but like a lot of space is booked up at InnoLabs, and always great to meet people. What about you? You went to a really cool event also in Long Island City / Queens.
Yeah, so a couple of kind of fun facts about Opentrons. So Opentrons did start out of GenSpace, and we talk about GenSpace a fair amount here because it's where we met, and I'm on the board of GenSpace. And the first office that Opentrons had is in the building that our office is in, 68J. They were on the 10th floor. Then they moved down the street when they grew to 20J, which is basically on the waterfront of the East River. And then they ended up expanding and moving to Inno Labs. We've had the original GenSpace founders, Ellen Jorgensen and Dan Grushkin, on the podcast. And then we've had two of the executive directors, Beth Tuck and Casey Lardner, also on the podcast. So GenSpace looms large, not only for Opentrons, but also for Curico. And we had Erica Milchek on our podcast not too long ago. Her company was incubated out of GenSpace as well. So after she left GenSpace, Ellen Jorgensen started another space called Biotech Without Borders, which is a community lab that is also in Long Island City. And I went to an event there. It was a biopunk event, and it was run by someone else who's been on the podcast, Elliot Roth. And Elliot has been doing a world tour, visiting community labs, looking for places where community labs can be built. And it's really an interesting movement, given this time that we're in that government funding seems to be drying up for research. There's a lot of people with lab skills that are looking for things to do. These community labs where you pay a subscription. I think Biotech Without Borders is $100 a month. They give you a space that you can go work at, you can go learn from. Elliot was presenting the space that he is responsible for or managing in San Francisco. I think there it's $350 a month, but they've done some really great things. And so Elliot was there to present what's going on at his biopunk space in San Francisco. There was a good crowd. Our friend Sabria, who's also been on the podcast, was there, Ellen as well, and had some great conversations with people. And very excited to see where things go with this kind of biopunk movement, community lab movement. I think it's super important. But then after that, as we've mentioned, we're going to London, and the craziness continues because when we come back, it'll be Climate Week NYC, which is basically 6 a.m. to midnight every day. I don't think we'll be on that kind of schedule necessarily, but there is this Venn diagram, as we've described before, where climate tech and biotech meet. And so there's a number of events that we'll be attending that week as well.
Because again, Climate Week, there's so many events and venture funds and community builders that create huge Excel sheets with hundreds of events that happen within those 5 days. And sometimes there's Sundays and Saturday events that are anchored there. And yes, you can fill up your whole schedule with events. And there's cool ones. Google throws some. And yeah, I definitely want to show up at a Google event because they're definitely well funded. But yeah, we're going to be hitting up New Lab. There's events that Deloitte's sponsoring. Everyone's in on it. And also the phrase that you were talking about that we've been borrowing from Kristen Ellis is that the Venn diagram between climate and biotech is a circle because it is the same thing. And that's why we show up at Climate Week, because we know that biotech has an absolute central role in climate solutions. And it must, because this world is a biological world. But we don't know what worlds are like in other planets. So that's a segue to the next topic.
Yeah, so we've got two quick science stories before we get into our guest. One of the things that we've talked about is this crazy comet that is entering our solar system. Erum, do you want to talk about this a minute? You're more the space person than I am.
Yeah, absolutely. Well, at first this came to my purview when I sometimes take 30 minutes to scroll on, I do reels, okay? And then there's this guy who was like, oh, there's this unidentified flying object in interstellar space that's heading towards our sun. And it's weird because it has a weird movement pattern. It seems like it's being steered and accelerated. Like the movement, if it was a comet or asteroid, would have been like, oh, there's some gravitational forces pulling it, and it would go like into these elliptical patterns and do slingshot traveling. But this is like no slingshot, just straight direction. And I'm like, what is this? And of course, I'm seeing the hyped up content and all the speculation. And this really stemmed from, there's this professor at Harvard, Avi Loeb. He is the Bard Professor of Science and the Institute Director of Harvard University. He's a best-selling author and wrote Extraterrestrial and Interstellar. And he's saying like, wait, let's just scenario plan this. Let's just game this out a little bit because this doesn't have a pattern of a comet or an asteroid. And so there's a couple things, and I need to do some more research, but stories are getting mixed up. You know, there's one called the 3-Eye Atlas, and this came to our purview because there was recent news that it turned green as it approached our sun. I think people are like, oh, why? And then I think what they're saying is because of cyanide. There's cyanide on this comet. Or like, wait, what?
Yeah, it's got this plume of gas, and apparently the plume of gas contains a lot of cyanide. And as it has gotten closer to the sun, it's turned green. And I looked at this guy, Avi's blog. It's really worth looking into if you have any interest at all in these unidentified planetary phenomenon. I know there's another name for it, but these huge things that are comets. And he even has a piece in his blog that says something like, if you want to do interstellar travel, you really want a bigger ship. And apparently this comet is humongous.
Manhattan sized. Yeah. So it's, hey, you know what? It's the end of days, Karl. Just with the news and everything, it's like, I would not put it past this actually being an alien spaceship coming to Earth. I would just be like, check the box off of crazy actual things happening in this world. I don't want to be a doomsday person. I'm just making some statements here that are for entertainment value only. All right.
Yeah. And I think of it more like the news is so crazy right now that Grow Everything is a beacon of sanity. And if it turns out that it is alien spacecraft, I'm all for it, unless they're here to destroy the planet.
Yeah. I'm interested in what's their biological makeup. Is it biology? What is biology?
Okay. So speaking of biology, just one quick story before we get into our guest. I flagged a story about octopuses, and apparently they prefer using different arms for different tasks, just like humans are left and right-handed. Octopuses have arms that they prefer to use for lifting things or pushing things aside or for hunting. I thought this was fascinating. I think the world of the octopus continues to surprise us. And again, it's like just another example of nature has all these answers and we just don't understand them yet.
Yeah, yeah. I love, you say octopuses, octopi, tomato, but I'm sure it has to do with their decentralized nervous system, I believe. So it's interesting because then it's like, do each of their tentacles have different thought processes? And because they're like trained to lift, they're trained to take something out, scoop things up and hug or whatever. So that's really interesting. I have such respect for cephalopods.
Yeah. One thing that's interesting about this study is they did find that they favor using their front arms. So front meaning in front of their eyes, with a split of 61 to 39% respectively, considering the actions that were being tested. So they tend to use their two front arms more often to reach, raise, lower, and curl. And they use their rear arms for sitting upright or moving like a conveyor belt, the way that cephalopods move. So anyway, continue to learn from these beautiful sea creatures and just more confirmation that the answers are in nature. We just need to spend more time learning.
I absolutely love octopi. I have such respect for cephalopods. I do not eat them. Sometimes I go to like Italian restaurants and I see octopus in the menu. I'm like, nope, no thank you, because I feel like they're super intelligent creatures. And I do think that they might be aliens from another planet, but that's just me. And that's not science. But that's that. So now on to worm news. We have the latest worm news, and you're tuning in to some of the best content about biology. So why are we talking about worms today, Karl?
Well, our guest is actually someone who works in the element space. And one of the things that we've learned through her is that humans and worms have co-evolved. There's billions of years of co-evolution that we're going to talk about. And they have basically optimized themselves for radically different environments than the ones that we live in. And I think this goes back to something we talked about many episodes ago when I said we need to have more dirty biology. Worms are like, they live everywhere. So they're the masters of dirty biology.
And so we got connected. One of her colleagues reached out, and Dr. Andrea Choe has a fascinating story. She is the founder, CEO of Holoclara, which is where she studies worms for their biochemistry and seeing what type of therapeutic molecules can be used for humans. She has her MD from the Keck School of Medicine at the University of Southern California, and she has a PhD in biology at Caltech. After we first met her, before we interviewed her, I was like, okay, even though we didn't know her for a really long time, I felt like she has been a friend of ours for a while. So with that, I'm super excited for you to listen to our interview with Dr. Andrea Choe.
All right, Dr. Andrea Choe, Welcome to the Grow Everything podcast. We are so happy to have you here today.
Thank you so much for having me. I am really excited to be here and talking with you guys.
You have such an interesting background. The work you're doing is so fascinating. You have gone from evolutionary biology research to building a biotech startup around, get this audience, worm-derived therapeutics. What you've done is anything but typical. What was a moment that convinced you to go on this trajectory?
Oh man, great question. I would say that once I made the discovery, which we'll get into at some point in this conversation, but once I made it, I actually went back to finish my last years of medical school. For anyone that knows the MD PhD program, you do your PhD. And then in the last 2 years of the program, you're in the hospitals. All right. So you wake up, you get to the hospitals 4:50 a.m., you talk to patients, you help with their care. And I would say while I was talking to them for those 2 years, sharing my story and what I had found, they had all just made the assumption that I would take this forward. They're like, oh, when are you going to bring this forward? When are you going to bring this to market? And then it wasn't like they persuaded me or I was coerced to, but every time I heard it, it had this resonance of truth. Like, yes, that actually is my path, right? So at some point when I finished my medical degree, I thought, okay, I've got to do this. And I just saw no other way. One of my earliest angel investors said that he invested in me because he got to witness seeing the phone ring and seeing me answer the call. So I don't know if that's too philosophical an answer, but really that was how it happened.
No, it's amazing. It sounds like there was a demand, right? And Karl teed it up, worm-derived therapeutics. So tell us about your discovery. What happened?
Where do I even begin? I would say that I walked into a worm lab. Let's start there. When you start your PhD, you walk into a variety of labs. I actually originally worked with mice. I worked with flies, and then I walk into a worm lab. And then from there, you can study anything that you're curious about in life. Think about all of the Nobel Prizes that have gone to worm scientists. It's because they can extract so much incredible detail about how life is programmed. So that's what originally brought me into the worm lab. And then from there, I just saw them engaging, interacting with each other, and I became deeply curious about them from like a animal behavioral perspective. And it's interesting because I once met Jane Goodall, and she said the very first animal she ever had, her first pet, was a worm. And by the way, my friends thought that I made up this story for years until like last month she went on the Call Her Daddy podcast and shared this story. There you go. But imagine you are a young Jane with worms in your hand. You become deeply curious about them. You have the patience to ask questions about them and how they might communicate with each other, which, by the way, you normally wouldn't think that because your experience is probably one worm on a sidewalk at a time. You don't see them engaging. But when you walk into a worm lab, you do see that. You do see them engaging with each other. And I could not help but become curious about how they're communicating without eyes, right? We don't have ears. It's got to be sensory or chemical. So I embarked on a journey to understand their molecular language and how they communicate with each other.
Yeah, wait, before we get into that, I just have two comments/questions. One is Nobel Prize winners for worm research. I had no idea. You mentioned that. And so clearly there have been Nobel Prize winners that have studied worms. Can you maybe tell us about one or two of those before we get into your discovery?
Yeah, understanding how RNA, how DNA is programmed in the worm, understanding the application of fluorescence protein, which was a tool that was then developed in the worm, helped us to understand a lot about how life is encoded, and also how worms can be used as tools to understand science, that you can now encode any gene to glow, right? So then if you become deeply curious about what is this gene that we found in the Human Genome Project, what does it do? You can actually tag it to a glowing protein that was derived from jellyfish and programmed in the worm to light up, which is a transparent organism. So now you could see where it lights up at any point in its life. So those are some of the discoveries that were made in worms. There's so many other discoveries that were made in worms too, not yet Nobel Prize winners.
And one other just kind of comment, Diary of a Worm. Have you seen this book? It's a kid's book, probably aimed at like pre-readers. Erum, you're probably at the point where Autumn is ready for Diary of a Worm. And there's a couple of them, and it tells the worm's story from their point of view, and it's hilarious. But let's get into your discovery. I digress. I'm sorry.
No, that's great. I have a 5-year-old son, and if there's anyone that should be reading it to their child, it should be me. So I will find it later today.
So tell us about your discovery and how that has led to Holoclara and what you guys are doing.
Yeah, so where we left off was that I became interested in studying how the worms were communicating. And the other thing is when you open the worm freezer, so there's a -80 freezer where you open it and the cold fog kind of drifts out. And then there are shelves in there, like at a library, and it's organized like the Dewey Decimal System. So then you can look for the worm species that you want, pull it out, and then there's a little tube that you can flick the lid open, scrape a tiny piece out, put it on a petri dish, and then you see them crawling within hours. And that could be any mutant strain, right? Or some species that you gathered from some other part of the world. And then someone could actually print it out and paste it in the room, an evolutionary tree of all these worms and how long ago they evolved, which blew my mind. I was like, wait a minute, this worm evolved 100 million years ago. This one was 200 million years ago. This one was 300 million years ago. How do you even know? And it turns out that because C. elegans, one of the organisms, was so well studied, people thought to study all of these worms around it and say, this is how far away it's diverged from C. elegans. And you can tell that by looking at the RNA and the DNA. So I thought that by itself was fascinating. So then I thought, wait a minute, I actually have these worms at my fingertips, and the ones that I cannot get, I'm going to email and introduce myself and say, I'm a scientist from Caltech. Will you send me your worms? And it worked. People sent me their worms from all over the world. People would gift me worms. I would come to work. I would see it on my bench. Here's some worms for you. And then what you could do is you could either watch them move around, but what I became interested in was collecting their secretions and then partnering with a chemist at Cornell who could do mass spec and NMR and tell me what was in them. So then from there, I originally intended to, publish like a worm book with chapters of it, like, here's what this one did and secreted, and this one secreted, and this one secreted. But ultimately, what we accidentally found was that they were all secreting something similar. There was some common denominator between all of them. And then when we found it, we said, wait a minute, we knew about this already. This is something that puts C. elegans into hibernation. This was what helped launch this whole field of longevity in C. elegans and aging. How could it be that such an important pheromone is used by other species? So then we started mapping it out and we saw these other patterns. Wait a minute, there is this one sugar chain that's not found in any other animal in the worm. It's like a world. It's not found in any other animal in the worm. See, now I'm just using the word worm to replace any word that starts with the letter W. And then we saw that different lengths of the tail, like 3 carbons versus 4 versus one with the carboxylic acid, they were like different letters of the alphabet. And then they all spoke the same language, which to me is mind-blowing because it's been this secret, invisible language that we didn't know that they were using, that they could communicate with each other around the world without us even knowing. So then that became my thesis, was the discovery of this sort of universal worm language. So that's also a whole thing on its own. And I could get to part 3 of where this brings us into biotech, but any questions there?
Yeah, no, this is fascinating. So you have this worm pheromone language. When we first were introduced to you, I'm like, okay, I'm curious. So tell us the work that you do. What would someone see, touch, experience if they were in your lab or holding one of your therapies that are in development? How did you make that leap from worm pheromone language to therapies?
Yes. So I would say the walking into the lab back then is a very different experience into walking into our lab today. Walking into the lab back then, you would see all of these flasks shaking with different species in them, me collecting all of their secretions. And then over there was mosquitoes infected with larvae. You could see the worms explode out of them. And then you would see, I had roses growing on the windowsill. And I actually planted them there when my lab mate left town. He came back. He's like, oh, nice roses. I'm like, oh, they're infected. They're infected with parasitic worms. So it was a zoo. It was for sure a zoo. And that's chapter one. Then after I discovered, along with our collaborators, this worm language, then I heard this Radiolab podcast about how there was this guy who had terrible asthma. He went to Africa to get hookworms on purpose. He walked around barefoot and he said, I'm going to get hookworms because I've heard that they can treat allergic disease. And then he treated himself and got better, which I thought was bizarre, right? Why would he do that? And then I just started looking online and finding that actually a lot of people with these severe diseases are taking roundworms, parasitic worms to treat their disease. So then that led me to try to figure out why is that and why would worms do that? And it led me down this whole path of realizing that worms are actually secreting something immunomodulatory. And then I thought, I actually think I have that. People are trying to figure out how do we get that therapeutic effect without having to eat worms? Do people know what worms are secreting? And my answer was yes. I think we found the Rosetta Stone of worms. So why don't we look there and try to decode this? So from there, we tested our molecules in animal models of disease, blocked a couple of diseases. And then everybody that I was working with was like, wait, when this should become a therapy, you should start a company. And so that's how the company arose. We took a molecule that we found out of worms, started synthesizing it from scratch. So now part two is without the worm, sans worm, the molecule in its purity, right? And then the cells that you're seeing mostly in the lab are mostly human cells because now we're testing them in that context. So now if you walk into our lab today, it's mostly like here are our T cells or our B cells or our dendritic cells or macrophage that we're growing and testing how our worm-derived molecule interacts with the human system.
Yeah, I have two comments. First of all, you said macrophage in such a classy way because we've been talking about it and we say phage, like just like hunky dunky people, like, hey, phages, phages. Okay, moving forward, we're saying phages.
Forever. It sounds like the yogurt. It's that Greek yogurt.
I was just going to say, my husband eats that yogurt and he was like, oh, I'm going to get my phage or phage. I was like, what is he saying?
Yeah, he's talking about phages.
And then that story about the guy who went to Africa and got the hookworms, I first read that in a book by Rob Dunn, who is an amazing biologist, and his book is called Wildlife of Our Bodies. And his approach was that maybe like hundreds of years ago, I don't know the correct timeline, we didn't have shoes. We were walking around the earth and worms were always around us and they were part of our ecosystems and had effects on us. And so whether they're in or on us, our bodies and our immune systems and other systems became acquainted with these other organisms. And we had talked about it several episodes ago because I was reading the book and I always like to recap some things I'm reading to make myself sound smarter. But it's really incredible to be able to see it because we live in such sterile worlds now that there's no way people are walking out barefoot near like a latrine, which we don't have here in our Western world in the United States, because you're in the US. Maybe we need to. And this guy did, and he cured a disease. It's like how much of our bodies today or these autoimmune disorders are caused by our sterile environments. And I thought it's a very interesting thought, which I am so curious to know if this is something that you and your team have been thinking about.
I think you've nailed it. The key there is to go back in time. Let's go back in time a little further from that turn of the century, right? From the 1900s. Let's go back further to the earliest human ancestors. So now pretend that you are an Australopithecus, you're friends with Lucy, you're on the grasslands of Africa, right? And there you and all primates are constantly getting roundworm infections from birth to death. So that's number one is you are right. You're acquainted with, you said that, they're acquainted with these roundworms for your whole life. Now, so were your ancestors and so are your progeny. So now let's do another shift in perspective. Now imagine if you're the gut worm and you're in the gut. Okay. So let's just stick with still being in the grasslands of Africa at the time. And you're sitting in the gut and you see a hole in the wall. That's a problem because you're there. You want to live in the intestines for not months, years to possibly decades. Okay. So you need to patch that up. Although there is no Home Depot for worms in the human body, right? You can't go buy a bucket of spackle and paint, right? So what you do know is that the human body already has their versions of that. And you can start secreting things and seeing if you could lure them in. So let's say you're a worm and you're sitting by this hole. Okay. So then you start secreting things. And then lo and behold, one of those compounds ends up recruiting neutrophils to come eat up the debris. And then another molecule, or maybe even the same molecule, better if it's the same molecule for efficiency's sake, can then also attract these epithelial cells to start laying down and replacing what was lost, and also stem cells to do that. And then now, don't forget about the plumbing, right? So then you need to call in the endothelial cells to lay in new pipes, the vessels, to bring in oxygen and nutrients. Bingo. So that's what you do. And you have all of time to figure that out. Then the other side of that is now you're sitting in your home and you're like, okay, job well done, worm, job well done, right? And then suddenly you see that the police force of that human ancestor is starting to come detect you, and it's blowing like shots at you, bazookas at you. And you're like, oh no, this is not good. And by the way, if it misses, it's actually creating another hole in the wall. So you're like, oh no, I have to deal with this again. So the easiest thing to do is now to secrete something to tell it to just chill out, quiet down, put away the bazooka. So that's their incentive. They need to do that to survive. So then you and I are sitting there thinking, oh my gosh, if we were playing this video game, I don't know how we would figure this out, but they have time. They can secrete whatever they want for years, decades, hundreds of years, thousands of years, millions of years. So they've really dialed it in. So now we're acquainted and we rely on that dynamic. And then we fast forward to the 1900s. Suddenly, the global north, the North America, Europe, we decide this is gross. We begin implementing an aggressive hygiene routine. We start removing worms. And lo and behold, we get all of these diseases, allergic disease, autoimmune disease. So is that a coincidence? Probably not.
Yeah, I mean, it's super interesting because parasitic worms is something that people maybe think about if they're traveling to a developing country. And maybe what people don't know is like there's thousands of species, correct me if I'm wrong, Andrea, and they range from microscopic millimeter size to like 100, a meter long sea worms. So there is like this range of worms, and maybe the ones that most people are familiar with are the ones that you see on your sidewalk that come up after the rain. And these are incredibly important for the soil. So we need them to churn the soil, digest the soil, keep the soil alive. And there is an issue with over fertilizing and trying to create these aseptic environments to grow food in. But when it comes to our own living systems, they're incredibly complex. And it seems like you guys have figured out like this language, but now you're working with these living systems that can be very unpredictable. What has been a setback or something that you've seen that has allowed you guys to leap forward?
Wow, you just said a lot there. There's so many interesting facets to your question that let's just unpack that. One is the unpredictability piece. I think that is such an interesting word because it's really in the eye of the beholder. I, in some ways, think that this longstanding relationship between worm and human has made this the most predictable relationship. Then we suddenly remove something and the problems happen. To me, the obvious solution is put it back. And then on the other side, we're so accustomed to wanting things to work in some unique one way that we have control over as humans. And then we get really comfortable with it because there's something about how that story is told that now it's like, okay, I've never had an mRNA vaccine before. Let's do it. So it's just that uncertainty is really interesting to me because I find that a lot of things that we do today as modern humans are uncertain and unpredictable. And then I guess the side of your question is, what did we learn? I think I learned to navigate that truly. I went into this as a scientist, as a physician scientist, right? I didn't go to business school.
You saved your money. What a waste of time. That's my personal thought, not a thought of everyone. So sorry for everyone that spent the money. I'm sure you have a great network of people.
Yeah, no shade on our friends who went to business school.
Not at all. But I think my only reason for saying that is I didn't go into this knowing originally how this worked, that you have an idea, that there are milestones and venture capitalists and they have expectations. So then learning that whole infrastructure and then having to slot into that was very interesting to me. And learning how this whole world functioned, which is primarily money driven rather than a lot of times it tries to go hand in hand with unmet need, right? But it's primarily profit driven. What is it that you're going to do that gives us this extra turn in this many years? It's not about a certain disease. It's like, do you know that those patients are suffering? Do you know that they only have 2 FDA approved drugs? Sometimes I hear like, oh, they're not really orphan anymore. So I don't know that we would get the extra incentive to make the extra capital to go into that. And then just being able to hear that time and time again, I will say it still surprises me every single time. Because where is the human curiosity to have discovered something that could potentially reverse the tides of disease? That's actually sitting in the hands of a funding framework that was very non-intuitive to me. So I think that the learning was that, one, I cannot rely on finding like billionaires that want to come find me and drink a glass of wine and talk about worms, right? I can't be in my own silo drinking a glass of wine thinking like, oh, that's that. I have to learn the model and I have to find a place for them to coexist. So I think that was just the greatest learning and probably is the greatest learning for any scientist turned entrepreneur.
Yeah, exactly. Oh my gosh, it's such a amazing point because, you're not talking about a setback that's scientific. There's setbacks you learn and then you adjust and you continue to move forward. The setbacks in the current capitalistic institution in which you have to operate. And something that we talk about, and we can continue talking offline, but just like what are new financial models to support science and entrepreneurship? Venture capital, is it the only way? It's a way that's been tested in other industries. Can we have new ones? But back to the work that you're doing, I want to get back to this idea of worm-derived molecules and how they can help with allergies and autoimmune disease. Clearly have discovered some molecules. You're testing in animal models now, but you're testing worm-derived molecules in worms, or you're going to mice. And tell us what discoveries you made, like specifically, like what specific autoimmune disease maybe are you targeting? We'd just love to know some of the progress on the science side at Holoclara.
For sure. So we actually tested them in animal models, some of our molecules in IBD, asthma, and for anyone that's trying to discover something, you usually stop at that point. You're like, oh, let's start a company. And I felt like there was something in me that was driving me to find another indication. In that world, they call it indication, is like the disease that you first go into. That's what you're asked. It's like, what is your first indication? And I do not know what compelled me to do this. Like I sometimes look back at my own journey and I do not know what compelled me to do certain things. Like I just imagine that hand from the Addams Family, like this hand on my shoulder, like just saying like, join the worm lab. Abandon your project and study worm communication. abandoned the MD PhD program. And I think there was this hand that told me to find this other disease. And that disease was eosinophilic esophagitis, which is a really interesting one because it's a little newer. It was, I think, maybe 15, 20 years ago is characterized. A lot of people that had trouble swallowing food thought, oh, I must have GERD, or I should just be chewing my food more. But then now they do biopsies and they look in their throats and they realize, no, you actually have a lot of inflammation that's making it hard for you to swallow because your esophagus is now narrow. And now they know that children get this and adults get this. And I've gone to these conferences, which, by the way, for newer diseases, right? I just said that this one was more new. I am so inspired by the people that come out. It's the parents that come out. They're asking questions. I want to understand how this works. They're driving this forward. So the patient advocacy groups for this disease, super inspiring. Have you ever seen what was Lorenzo's Oil? That movie? Do you remember that movie? Back in the day?
Yeah.
It was the parents that found the solution for their child. Going to these meetings, I was just really pumped to try to help this group of people. They would tell me stories about how their kids can't have cookies, and then their sibling gets a cookie, and then they feel so guilty. They go to the bathroom to try to eat it privately, but then they come out and they start crying because they did something that is just like the stories of how these families are affected. It's not just the kid that has the disease, it's a whole family. So that one, I felt really pumped to try to find a solution for them because also the kids don't have that many solutions. There's only 2 FDA approved drugs, and one of them is a steroid, which kids cannot take. So then they have to get an injection every single week. So can we find an alternative for them? And when we actually went up to Glenn Ferruta, who's one of the amazing, they call him one of the godfathers of EOE. And I asked if he would study this in his laboratory. We studied in his lab and was able to block the disease there. And so that's part of the journey that we're at now is, okay, let's help these people. And our first molecule, we've already taken it through phase 1 clinical trials. It's cleared safety hurdles. People have ingested it. It's that we found no significant adverse events. We've given rodents 10,000 times the therapeutic dose and could not find tox. I think it's just about getting it now to patients.
Let me just ask, let's also put this into a different kind of perspective. When you've gone to these meetings, this is a relatively new disease. Like, how many people show up at these meetings? You said a lot of it is the parents. Is it 100? Is it 1,000? Is it 10,000? Just give our listeners a sense of the scale.
I would say it's around 1,000 people. There's a West Coast one and there's an East Coast one. There's 2 advocacy groups. So I would say it's cozy. And I actually like this because I've been to a lot of conferences where there's like Society for Neuroscience, right? There's just tens of thousands of people. But this one, you see everyone, you know their names, you know the players, you stop them in the hallways, you talk to them about their research. You see patients that ask questions, you can go up to them afterwards and you go on a walk with them through a park and talk about the disease and their family and everything. So I would say it's in that range.
I think that's great. For me, it's just 2 things. One is like you get to know this community where you're making a difference. And 2, because this is a new disease, it's probably still considered a rare disease and it only has 2 approved therapies, one that children can't take. It means that the impact that you can make can happen a lot faster.
Yeah, it's like a pilot indication.
Exactly. Then the other question is, when you think about the community of people who are doing worm pheromone research, like how big is that community? Is that also 100,000?
That is something that I have wondered about for a long time because after I heard the Radiolab podcast, I actually went into the literature to figure out who's studying this kind of stuff. It's a whole field called helminthic immunomodulation. And most of them are in Europe, and I do not know why. And then also they discover these interesting compounds from worms from different species that do amazing things, and then they don't do anything with it.
That's crazy. Oh my God. VCs listening. We've got several dozen company ideas coming.
Yeah, absolutely. And our friend Benji, who does like longevity drug, not discovery, but experiments and has a whole, I don't even want to say like patent coins. We just interviewed him. And basically they do experiments you can watch, and then people can put money into getting these drugs validated to animal studies. And then when they sell the drug, the people that put money in will get money back. And that's all I'm going to say. That's how it works, but I don't want to get too crazy.
Yeah, but Andrea, I think it's so fascinating. What did you say, Helminth? What's the field called?
Helminthic immunomodulation.
Helminthic immunomodulation. Okay, everyone, listen to Grow Everything. This is probably the heaviest part of this podcast. Just knowing that name.
There are incredible scientists out there that are studying these species, which, by the way, now we go and get the species, sometimes from those authors, right? We find them, we partner with them, and then we bring them in and we study them. And one of those is Rick Maisels, and he's in Glasgow. And I have looked up to his work for a very long time and asked him to help what we're doing, join us in our efforts. So he does work with us, and he is a part of Holoclara. So that's an example. And he's in Glasgow, right? So there are people around the world that are doing this.
Yeah, the other message that is coming loud and clear, and we say this a lot on the podcast, is that all the answers are in front of us. We just haven't uncovered them. And there's a lot of them. And so you're an example of, you're looking at something that most people wouldn't ever think has any kind of therapeutic effect. And yet all of a sudden, it's like there's this evolution, this coevolution, this symbiotic relationship that developed between these organisms and humans that we've just ignored and have actually antagonized. But right now, where you are in this business, give us a picture of where the business is, where's Holoclara, and what's the biggest challenges you're facing? Are they scientific, regulatory, cultural? You've given us a broad picture.
I think that right now we are really focused on advancing our lead molecule into a patient population. So that's what we're looking forward to. And we're really excited about our safety data, just clearing that path for us to move forward. The hurdles, I would say, I don't have any regulatory hurdles because, like I said, we gave rodents 10,000 times the active dose and didn't find any tox. So as long as you're safe, you're good. I think the cultural hurdles, for sure, they are there because, again, you just need to fit into that model. There's not a lot of people that are discovering natural molecules and pushing them through. What people typically do is you have a target in a cell that glows, and then you're put in a petri dish, and then you screen thousands of man-made drugs to say, did that tinker with that signal, right? And then you take it forward and pray that there's no tox. And a lot of times there is, and you don't find that out until you're in your patient population. And that to me is really interesting because you're not looking at everything else that it's touching upon because you only tagged that one protein with a glowing signal. So it's almost like, I just know that it's working on this target. I'm not going to look at anything else. And that, for some reason, gives people the sense of safety that they can go to sleep at night. But that's a myth, right? That's not how things work. So now that we find natural molecules, instead of just marching forward with it into clinic, people expect you to backfill every single thing from the beginning, right? And then for people like me, you have to do both simultaneously with half the money, because then you've raised money to go into trial. And then the expectation is, but just magically figure everything else out at the same time based on these milestones in front of you. So I think that it's easier to do all of that than change someone's mind. Like we will just go figure everything out with the fraction of the money, with a fraction of the time. That is easier to do than changing someone's cultural expectation of how to move forward.
You've definitely touched on my own personal frustration with the pharmaceutical industry as it is today. I worked in pharma early in my career, and I left because I was frustrated, because it's like, wait, what is going on? And it's like so stuck in its ways. It's so rigid. But I sense a disruption happening. I said it before. I think pharma's ripe for disruption, but there's also disruption happening, whether we like it or not. Perhaps in the world of FDA, there's a lot of question marks going on. Will it be a good thing or a bad thing? And then there's the two worlds of pharmaceutical versus nutraceutical. And it's like one's underregulated and one's overregulated. And so it's like, okay, like what gives, right? Like, and then my whole thing is, can we just live in nature and eat fruits and vegetables that are grown,
Oh, I am with you. So I actually thought about going into the nutraceutical path, which is why I recruited Peter Barton-Hutt to my board, because he was former chief counsel to the FDA. And he's helped establish this whole generally regarded as safe pathway. What changed my mind, though, was reading a study that a lot of people do not want to take drugs unless they are insured, FDA approved, covered, and that they can have a $2 copay instead of paying for a nutraceutical that might be $20. And to me, I think that's crazy because I would pay that amount of money for my life and my well-being, but it's also just an expectation, right? I am used to paying $10 for my drugs, and a lot of people can't afford more either. So then understanding that system, knowing that there was a study where the copay went up by like a couple of bucks and people just decided to get sick instead. So then I thought, okay, if I was really going to make an impact, I do need to go down this path and have an FDA approved payer covered drug.
But that doesn't preclude you from potentially creating an ingredients company separate that is creating ingredients based on this research that could be added as a supplement to foods. I don't know what that looks like. I'm just throwing it out.
Oh, totally. That's like if I find that billionaire that wants to come drink wine and talk about worms, let's do it. Then we start the whole pharmaceutical company based on worms, and it's a nutraceutical pharmaceutical company where we can help lots of patients in need, but also develop nutraceuticals too.
Let's jump to that question. If resources and technical limitations disappeared tomorrow, what would your dream experiment or moonshot be for Holoclara?
I think it would be exactly that, is getting all of the species, which by the way, we do have a lot of them, but now having just the time, money, the team, the talent to characterize the worm atlas of interesting molecules, take them forward into different indications, finding the people that need them and getting it in front of them. It's so funny because the technical limitations is not what I lose sleep over at night. I feel like we can solve for anything. So as long as we have the time and money and team. So I think that would be the goal.
Yeah, sorry. I was just going to say, I just did a quick search because I was like, how many worm species are there? And my AI says there's no definitive number, but it's at least 20,000 and potentially 250,000. So there's a lot of worms out there that we can study.
Yeah. I have another question. Maybe we want to put this up top because it's like not the vision question that you just answered. But so worm-derived molecules, you're finding these and like they're natural. And I think one of the things that why pharma does man-made is because they could patent those molecules. Are there issues with patenting naturally derived molecules? How do you navigate that? Or do they change them a little bit?
Yes, so there's different ways to patent natural molecules. You could patent method of use, right? And you could patent formulation. You could patent the synthetic method, right? But also, I like to deal with small molecules. So there's small molecules and there's proteins. And I like to deal with small molecules, which, by the way, any brilliant chemist can modify those to create composition of matter. And sometimes we do need to, because the drugs are not appearing from a worm that's living in your gut. Now you're taking it orally so you can make a small chemical modification for absorption and permeability. And then you have composition of matter. So I think that there are solutions to that work for both, for everyone involved.
Oh, no, thanks so much for clarifying. Oh my gosh. A lot of different worlds we can go down in this conversation.
Hi, a quick shout out to our sponsor, Messaging Lab, the force multiplier for biotech. Your biotech company is making the world a better place. You know that, we know that. But does the world? There's a big reason why some biotech companies attract investors, sign up customers, and get attention. That reason is strategic communications. At Messaging Lab, we translate complex science and economics into compelling narratives. And we have done that for the most successful biotech companies across healthcare, agriculture, personal care and beauty, materials, and the list goes on. We're here to make sure your ideas not only get heard, but resonate with your audience. So if it's time to amplify your company's voice, visit messaginglab.com to explore how we can elevate your story and grow your business.
So, Andrea, before we go to quickfire, was there anything we haven't covered yet that you think we should have?
Oh, I think this is a wonderful conversation so far. You guys have really thoughtfully covered every part of the worm corners here.
Exactly. All right. The most surprising fact you've learned about worms that blew your mind.
Similar to what Karl mentioned about the many species, they are actually the most abundant animal in the world by number. So you can look it up right now anywhere. You're like, what is the most abundant animal in the world by number? It is roundworms. There are 10 to the 19th. I can't remember the word for that. I can't even remember the mathematical number for 10 to the 19th, but that many worms. If you erase everything in the world, you would see an outline of worms. So they actually do dominate this world. And I know that makes us uncomfortable, but they do.
Wow.
Holy cow. Yeah, that's crazy. Okay, what's one immune-related myth you wish would disappear forever?
I think this concept of it's like, okay, if you understand the one target, then you understand what it does and I can go to sleep at night. That's not at all true. This control-driven way of viewing drug development, like it's mythical. The drugs are doing all these other things. If we can just try to really accept that we've co-evolved with worms and they're secreting molecules and your ancestors have already been exposed to them, you need them. Those would probably go hand in hand with how I think about myths in immunology.
Amazing. Amazing. Okay. If you could borrow one superpower from the worm world, what would it be?
Hibernation. Don't you? Wouldn't you want to just go to sleep until things are better?
Oh, yeah. I would want to dig into the ground and go like far underground and see what's going on.
I love that. That's the opposite of flying, right? It's like people are like, oh, I want the superpower of flight. You're like, no, I just want to dig.
Yeah, I want to go and see the different layers of the earth.
You could do that in Minecraft today. You don't need a worm superpower.
Okay, well, I'll have to play that when my son's a little older.
Okay, so Andrea, one word to describe the future of worm-inspired medicine.
Oh, that's a good one. I would say balance. Because we live in a world today where we just want to wipe everything down to zero, get rid of all the eosinophils down to zero or this cytokine down to zero. But really, it's about living in harmony and in balance. And I think that we have been in balance with worms for a very long time, and then removing them created an imbalance. So how do we restore that balance? I think that we could do that.
Beautiful.
Yeah. Andrea, this has been such a great conversation because we touched on so much. First of all, talking about worms is not something that we do every day, but now I think it's been added to our vocabulary. Two, what you're doing is inspirational and just opened my mind to so many possibilities and just reinforces to me again that every answer that we seek is right in front of us. We just have to uncover that. And this whole, drug development is extremely complicated and you're challenged with that, but also overcoming it. So congratulations. We're so happy you came on the podcast.
Oh, thank you so much. I really appreciate the thoughtful conversation. I know we don't have wine right now, but it does feel like we sat around the fireplace with a nice glass of Malbec and was able to just explore all of these really interesting topics about past, present, and future. So thank you so much for having me and joining me at that level of conversation.
Yeah, it was great. You're also very easy to speak with. And I will say, if there's any billionaires out there, we have a glass of wine waiting for you. Call Andrea.
Yes, there you go. I love it.
Thank you. Thank you.
Erum, what did you think of that episode with Andrea? I have so much to say because she made me feel so smart.
Yeah, and she made me feel so friendly. Hello, like you guys heard it. Andrea Choe is such a cool person. She's very down to earth. She's very warm, as you can tell by the work that she does, her focus now, working with this patient advocacy group. It was great to see that she was going down this path of becoming a doctor, and she saw this opportunity and she saw that worms hold an answer, but there wasn't a lot of exploration in looking at worms for the therapeutic purposes. And she went for it, which is amazing. So tell me, Karl, what did you think?
Just this idea of using worms to treat diseases. I know that worms have been used, maggots have been used for people who had gangrene. They're still used medically. I heard a story from Elliot Roth about a team of researchers working out of the Biopunk lab that had taken maggots, crushed them up, and applied them to Band-Aids, and they found that wounds healed more quickly. So I think there's a lot to be learned there. And I'm really excited about the work that Andrea is doing, because worms, parasitic worms, as much as people try to avoid them in certain parts of the world, people are infected by them, but they do have these immunomodulatory effects that we still don't understand. And Andrea is working on understanding that through her work at Holoclara.
Yeah, and it's very inspirational because, again, this is another example of how we can look at the large, vast world of biology that's outside of the human realm and see how it can help with humans. She's talked about how we evolved with different worms and how the worms became a part of our external immune system. I don't know if that's a term, but I just coined it because, hey, look, there's a lot, like you talked about, living in these sterile environments. Did we just chop off our external immune system? And that's what's causing all these allergic reactions. And the hypothesis is that if we create an environment that has different diseases, I don't know, and what's out in the forest where we lived for hundreds of thousands of years, 250,000 years for Homo erectus, that is, let's reintroduce that. And we talked about before when I had gone to that workshop of computationally designed organisms, the question that Joshua Bongard, who's a professor at the University of Vermont, said is like, what if a building had an immune system? And then how does that help us? And I think it's a beautiful thing that Andrea just took the time to respect the biology of worms and did this vast research and said, hey, look, there's so much here. Let's start mining. And I don't want to say mining. Mining is such an extractive process. It's just to respect Andrea and her practice.
It's much more learning.
Yes, more learning, respect, appreciate, discover. And then what will that tell us? She really talks about this communication, this worm language. And it's, I think, like in our circles, so I'm going to say this is like a popular thing, but maybe not to some, but if you're listening to the Grab Everything podcast, it could be to you, is that we're learning about how other organisms communicate. So people are looking at plant communication, specifically trees communication, the mycelium networks underground, and how that actually has specific languages and specific information. We talked to some scientists who we're going to have on the subsequent podcast, so stay tuned for that, about peptides being a communication molecule to tell cells how to behave, which is fascinating. And I'm borrowing that. You guys don't even know about that because that's an interview coming up. But I thought it's a beautiful thing. It's a beautiful thing to learn the language of biology from different organisms and apply it to our experience, our human experience.
Yeah, and to me, it's like just another confirmation that nature holds so many of the answers that we're looking for, and oftentimes right in front of our faces. And I just love the interview with Andrea, and I'm really looking forward to having her on again to see what kind of progress she makes, because she just blew my mind. When she first talked to us about the language of worms, I was like, wait, what? Does this fit into grow everything? But it does. As we got to know her, we just felt that what she's working on is so interesting and so important that we knew our audience would want to hear about it.
Yep. So stay tuned for more helminthic immunomodulation news. And it's been great. So we're excited for some episodes coming up. You're going to hear from Alessandra Zanari, Chief Scientific Officer of OneSkin, which is more about like longevity of skincare. Phil Morrill, who's going to be talking about bioeconomy in Australia, specifically how they work to orchestrate business deals, which is super interesting. Karl gave me a little bit teaser. I wasn't able to interview Phil, but you did, and you told me about how amazing it was. I can't wait to listen to it. And then, of course, our 150th episode live from BioFab Fair, hosted by Biofabricate. Just stay tuned. So much information about how biology is changing our world.
That is great. So with that, let's say end scene. That's the pod.